After listening to a lot of the biological talks the last couple days, I realized that in comparison, I think these wood cells are actually a lot easier. So I'm going to kind of have two parts of my talk today. In the first part, I've gone through, so I've been at this for about 10 years now, and I spent a lot of time in the beginning trying to develop methods to indent the wood cell walls and really understand the measurement. So I'm going to go briefly through a lot of that work. And if you want to, there's a lot of references in there. If you want to talk later, I have about 100 auxiliary slides from other talks I've given, getting into those details. Then what I really want to get to is some of the ways that nanoindentation has been helping us in the Forest Service and get us some of the materials research projects that we're working on. But first, I want to talk about how a research materials engineer actually fits into this mission of the Forest Service, which is to sustain the health, diversity, and productivity of the nation's forests and grasslands, and also to meet the needs of future and present generations. So one of the major problems, or one of the major issues facing the Forest Service and the forests in the United States is the threat for catastrophic wildfires. So you can see this map up here, especially out west. There's a lot of regions that are under a threat for these catastrophic fires. And the reason is because there's an excess amount of biomass that are in the forest. Out west especially, there's entire mountains of trees that have been killed by pine beetle infestations. And what happens is this material dries out, you have a spark, you have a huge wildfire, and the wildfire kills and burns everything. But then what will happen is the trees will come back eventually, but they'll be really close together, and then they'll get to be about 20 feet tall, half the trees will die, and then you have another forest fire comes through and kills all the trees again. So it's a really bad cycle, and it's a problem. And so as a materials researcher, just to give you a little bit of an idea of the approximate treatment numbers and the extent of the problem, there's about 400 million acres in the United States alone that are in need of some sort of restoration. And the Government Accountability Office has estimated that this would cost $69 billion. Currently, the Forest Service, we get about $0.3 billion a year to help with restoration efforts. But really, inaction is just not an option. The costs are rising in addition to the devastating losses and lives and property. It costs the United States government about $3 billion a year to fight these wildfires. So it's really a big problem.
Can I just ask, why do the trees grow back closer to each other after a fire?
Because there's nothing there. And so you have all the seeds next to each other. So the saplings can grow very close together. But then as they get bigger, they crowd each other out. And only the survival of the fittest, and so then only the tallest trees survive and the smaller ones die, and then you have this excess biomass problem again. And so the way to fix that is actually go in and mechanically remove the dead material. But that costs money, of course. And so as a research materials engineer, what I'm trying to do is I'm trying to find some sort of value for this biomass that's being removed to try to accelerate forest restoration. And so even if you can't recoup all the costs, if you can recoup half the costs of restoring forest, then you can restore twice as many acres. So that's the idea behind accelerating the restoration. To do this, we either need to create new products out of the woody biomass, such as biofuels or these cellulose nanomaterials, or we need to increase the demand for current wood products to create more market-based value. And so what I've been working on is trying to improve the durability of current forest products. And so that includes the dimensional stability, especially in relationship to adhesive bond line durability. So in wood swells, that adhesive bond line needs to be able to accommodate that swelling. Then other durability issues include corrosion of metal fasteners in wood, and also fungal degradation. So I'm going to get into that in a little bit, kind of the mechanisms responsible for the durability issues and how nanoindentation can help us understand and improve the durability of forest products. But it's really kind of the unknown cell wall structure population shifts that are really kind of hindering researchers' efforts in this area. So just briefly, what is this? I think everybody's here is aware is a nice anisotropic multi-scale material. And so the structure here is actually a softwood, which is the most commercially relevant species in the United States. That's why I'm going to focus on softwood in this talk. So the three main planes of structure are defined by the growth rings in tree. So you have the transverse section, the tangential longitudinal, which is tangential to the growth rings, and then the radial longitudinal, which goes from the pith up to the bark. And in terms of the cellular structure in softwood, the most common cell is called the longitudinal tracheid, which is kind of a banana-shaped cell that's about 10-15 millimeters long and about 50 microns in diameter. That goes up and down the stem of the tree. And then two of the primary functions of wood in a tree, of course, because the tree doesn't grow the wood for our use, it grows it for its own use, is mechanical support and also transport of liquids. And so the liquid can be transported up and down the tree through these longitudinal tracheids. And there's also pits, which are little openings between cells to allow that. And then also to facilitate transport from the pith out to the bark, there's also these organizations of ray cells, which are similar to longitudinal tracheids. They're flipped 90 degrees. So in terms of these cell walls, they're multi-layered materials. And the largest layer is what's called the S2 secondary cell wall layer, so the S2 layer. And this is basically a fiber-reinforced composite of cellulose microfibrils that are embedded in a matrix of hemicelluloses and lignin. And the defining feature of the different secondary cell walls is the angle that these cellulose microfibrils make with the longitudinal axis. And so in the S2 layer, it's a fairly small angle, typically between about 5 and 10 degrees in mature wood. But in these S1 and S3 layers, it's a much higher angle. So you can imagine that as the S2 layer swells, that would provide kind of some hoop constraint for the swelling of the S2 layer. And then all these cells are held together by a lignin-rich material called the middle lamella, which kind of acts as like a glue to hold the cells together. So we get going on a couple more levels of structure. So wood is composed of polymers. So the cellulose microfibrils, that's kind of the reinforcing, and like these are like semi-crystalline in terms of mechanical properties. The longitudinal modulus of these things is over 100 gigapascals. And the transverse is somewhere around between about 20 and 30 gigapascals. And then the xyline and glucomannan are different types of hemicelluloses, which kind of branch polysaccharides. And then the lignin is kind of this encrusting aromatic polymer. But all these polymers, they all have hydroxy groups. And one of the fundamental issues that lead to durability issues is the absorption of water. And so water can exist in wood. There's actually two different phases. The first is free water, so that's water inside of the kind of the micron scale cavities of wood, such as the lumen. And then the other form of water is bound water, where it's held by intermolecular attractions within the wood polymers, like at the hydroxy groups or different favorable sites on the wood polymers. And it's known that wood is a function of humidity, absorbs water, and the moisture content is defined as it's the weight of water divided by the dry mass of the wood. Can go up to about 30%. And so below this point here called the fiber saturation point, all water in wood is held bound inside the cell wall. And it's not until you get above this fiber saturation point that you start to get free water that forms. Now, so of course, these cell walls, especially in late wood, are about 5 microns across. So that's a good size that we can use nanoindentation to test the mechanical properties. But the wood structure isn't exactly the nice homogeneous material that a lot of the standard nanoindentation techniques were developed for. And what can happen, and one of the things we realized early on is that if you prepare an unembedded piece of wood, it's possible that when you perform the experiment, you can have some specimen scale flexing. And then the other issue is that when you're performing these indents, you're always performing them next to either edges, so free edges between the cell wall and the empty lumen, or the edges between the different layers within the cell wall. So back in my PhD at the University of Wisconsin with Professor Stone at the University of Wisconsin, we developed what we termed the structural compliance method. And basically what we discovered is that both the effects of specimen scale flexing as well as edges, they can be accounted by including a structural compliance into the analysis, and that the structural compliance actually behaves similar to the machine compliance. That is, it's independent of load. And so you can use the kind of standard equations that people use to account for machine compliance to also account for the structural compliance. And one such way is this modified Stone-Euler-Sparrow correlation. This equation is easiest to use if your hardness and modulus are independent of depth. And then if they are independent of depth, then a plot of the total compliance times square root of load versus square root of load will give you a slope it would be a straight line fit, then the slope would be the structural compliance, and you can just account for the structural compliance to correct for your low depth trace the same way you would a machine compliance. And with this method, you account for specimen scale flexing, free edges, header phrase interfaces, and also particles inside of the embedded inside of a matrix. And then, so in case of a thin film like this, this assumption that hardness and modulus are independent of depth wouldn't be valid, but you can still use a variation of this to account for a free edge and a layered specimen as well. And all these full details are here in these papers, and I'd be happy to talk to you afterwards as well. So the other issue with wood polymers is that they're polymers, and so the materials are really going to be time dependent. And one of my goals has always been to really develop nanotation into a local probe for mechanical spectroscopy, one that really provides insights into the underlying mechanisms that are controlling the properties. And I've always wanted to do this with a minimum of assumptions and just collect the data and interpret the data interpret the results. And so kind of to give you an idea what I'm talking about here, here's like a typical, how like viscoelastic data look for like a typical amorphous glassy polymer. So polymers, we have polymeric chains with side groups. So you can have like secondary relaxations where the side groups begin go through a thermal transition, and that does typically show up as like a very small little perturbation in the storage modulus, but you can get like little peaks in the tan delta. And then of course, as you go to higher temperatures, the entire backbone goes through a thermal relaxation, and that can cause a large decrease in the elastic modulus of the material, and also a large peak in the tan delta. And there's different ways to define a glass transition. One is like this onset glass transition, which would be important for like engineering materials. If you have a material and you want it to maintain its mechanical properties, you don't want to go beyond a condition where you start to go off this peak. But in terms of like the molecular scale dynamics inside of a system, oftentimes like the peak of the glass transition is what's more interesting. So I'm going to develop nanotation protocols to try to, by testing kind of bulk polymers with known behavior, that you can do conventional measurements on. So here's a set of materials that I started to work with. And so these are all the conventional viscoelastic moduli. This PMMA and low density polyethylene here, these were experiments that were tested using broadband viscoelastic spectroscopy in Professor Lakes' lab at the University of Wisconsin. I got pieces of those material from that study. And then these polyethylene, polyethyl ether ketone, and polycarbonate, and polystyrene were materials that I required. And then I did a conventional single cantilever test using a standard DMA setup. So one of the first ways I started to sort of think about how can we measure time-dependent properties. But also when I started this, there's a lot of literature that said if you change the loading protocol when you test the polymer, you get different answers. And so if you get different answers, then how do what does it mean? So I went through a very careful study where I systematically varied the loading time, hold time, and onload times. And I saw that indeed, so I calculated effective modulus in the usual way, the initial unloading stiffness divided by square root of area, and then using the standard Hertzian contact equation to calculate an elastic modulus. And then I estimated frequency as just 1/2 pi times the time of the unloading. And when you do these experiments, carefully, one of the things you have to make sure is that the unloading slope is actually an elastic response, so that you don't have some forward viscous deformation happening. But if you go through and carefully analyze the data, what you find is that systematically, these nanotation results, they're always higher than the conventional analysis. But what's interesting here from the aspect of trying to understand, are these mechanical properties related to microphysical processes, is that the shapes of the curves are quite similar. So there's, of course, another way to try to measure the viscoelastic moduli, and that's to do these dynamic tests, where basically you load up the indenter and then apply a sinusoidal load to it and measure the load and displacement, and you can calculate a storage modulus that way as well. And we do that on these exact same experiments. Again, we see that it's always systematically higher, but we continue to capture the shapes of these curves. So another thing with the dynamic notation is that you can also easily calculate a tan delta. And the tan delta is related to the shapes of these curves. And that if you go through all these materials, you also see that the tan delta actually lines up pretty well. So this is kind of gaining confidence that we're potentially making physically meaningful results. So now if we go to another parameter space where we start to vary the temperature, as we do conventional. So these solid lines here, these are conventional single cantilever DMA measurements, and then these the points here, these are the nano DMA3 measurements. And again, we find that it's always systematically higher, but the shapes are similar. So for instance, you can see this green curve here is concave up. This blue curve here is concave down, which is similar. And again, if you go to the 10, all right, so you could also plot this data as a function of temperature. And you can see that kind of onset TG lines up pretty well between the nanoindentation as well as the conventional DMA. And then also, if you look at the tan delta, you can see that the peaks line up pretty well. And so on this cast acrylic and the conventional DMA, we saw a peak at less than 1 hertz, and we see that same peak in the nano DMA3. And then the peak related to the beta peak, in the conventional analysis, that showed up the same place you'd expect. The nanotation showed up the same place it showed up in the conventional measurements. So this is really strong evidence that we can use nanotation-based mechanical spectroscopy to investigate actually glass transitions in wood cell walls, which I'm going to show later, that there's a moisture-induced glass transition in the hemicelluloses that we can actually detect with nanoindentation. And then finally, one other thing, the thing that I did back for my PhD was I'm also looking at viscoplastic properties. And there's some debate about what a hardness measurement means in a structural polymer like these. So what I did was I carefully measured Meyer hardness as a function of indentation strain rate by loading up the indenter and then watching how the area grows at a constant load. And then you can calculate Meyer's hardness as a function of indentation strain rates. And then using this 50-year-old Tabor-Marsh-Johnson correlation, you can convert the hardness to a compression flow stress like you'd measure in a uniaxial test. And if you go through this analysis, you can almost exactly reproduce what you get in the conventional uniaxial test. And this work was built off of a lot of previous work done in Professor Stone's group to develop a creep analysis. All right, so now getting back to, we spent all this time developing these tools. Let's do some interesting wood science with them. So one of the areas I'm really interested in trying to understand and trying to improve is wood adhesive bond lines. And so when wood gets wet, it swells, and you can have a strain mismatch at a wood adhesive interface, and then that can lead to failures. So how big of forces are we talking about in wood? Well, the ancient Egyptians have known for a long time that if you put a slot in a rock, put a dry piece of wood in there, pour some water over it, the swelling forces are actually large enough to split the rock. So these are pretty large forces that have to be accommodated at the wood adhesive bond lines. And we've also done some work looking at twisting in like thin wood slivers, and we built a little device to actually measure the torque that's produced by these wood slivers. And that we find is that on a specific torque basis, as a function of humidity, these wood slivers, they outperform some of these recently reported carbon nanotube torsional actuators. So nature's still doing a better job than we can do synthetically. So that's more motivation to really study these materials and understand them, because there's still a lot we can learn as material scientists from nature. So getting back to our wood adhesive bond lines here, so there's kind of two ways that the strain mismatch can be accommodated. One is that you could have a flexible adhesive, but I don't think you want to build a house out of something with a flexible adhesive that's going to creep over time and fall apart. And so the way that more structural adhesives we believe work is that they actually stabilize the wood surface. And so when you go down to that interface, you're down there at that cellular level, And it's believed that adhesives actually need to both penetrate into the lumens next to the surface and also infiltrate into the wood cell wall properties. So one of the best adhesives right now is out there is phenylformaldehyde. It's been developed over decades of trial and error in industry, and it's the best moisture durable wood adhesive we have available right now. So what we need is we need tools for cell wall characterization, and we're going to try to understand the mechanisms that cause or that result in the moisture durability. And the goals are, to both improve adhesive performance of current wood adhesives, but also if we can understand the mechanism better, that'll accelerate the development of more sustainable adhesives that can replace, these phenylformaldehyde adhesives.
Sorry, one question regarding how big the swelling forces could be. This swelling is based on osmosis, right?
It's absorption of the water in the wood polymers.
The mechanism of the force is osmotic force, or is it?
That's actually a good question. I'm not sure. Yeah.
It could be,
Yeah. So to develop techniques to be able to quantify how much adhesive is going in the cell wall, that actually led me down to the advanced photon source up at Argonne National Laboratory. And I guess I should also mention that I've been a resident user up there now for the past year and a half, so I've been using a lot of their tools and applying it to wood science research. So in the kind of following slides, you'll see some of the results of some of that work. But this tool, we took a bromine-labeled phenylformaldehyde. We made wood adhesive bond lines. We cut a 2-micron thick wood section, and then this X-ray fluorescence microscopy, which is done at APS Beamline 2 IDE. And this was the Beamline scientist, Sophie Charlotte Gleber. And this is Chris Hunt and Chuck Freihart and Daniel Yell, who are fellow scientists at Forest Products Laboratory, who are really chemists and were able to make this bromine-labeled phenylformaldehyde and also really understand wood adhesion well. So this technique, you have a 200 nanometer beam that you can raster over top of this 2-micron wood section, and then from the fluorescence signal, you can actually map out and very accurately quantify the amount of bromine that's in the wood cell wall. And then because every link on the adhesive polymer had a bromine, that's directly related to the amount of adhesive that's in the wood cell wall. And then what we do is, so we cut this 2-micron section. That means you had the wood block underneath, and I can go to that wood block and do the nanotation on the exact same wood cell walls that I took this section from. So it's the basic procedure I used. So the surface was prepared exactly from where that 2-micron thick section was. Nanotation with the Hydrotron tribodenter with the Berkovich probe. Then we're going to control the humidity inside the enclosure using an RH generator, and so we can look at the effects of moisture-induced softening on the cell wall properties. Just at room temperature and the multi-load indents to account for the potential effects of edge effects and specimen scale flexing. So here's just an AFM image of some of the residual indents that we placed inside these cell walls. So we purposely use latewood because it has these nice thick cell walls that we can easily place the indents into. And then so here's an example of a multiple load indenting function, and we controlled the load such that the depths were basically the same between the dry and moist environments. And just from these indents, you can tell that there's a lot of moisture-induced softening that happens as you change the humidity in wood. Then we were able to find these exact same cell walls back on the bromine X-ray fluorescence microscopy image. And just to give you an idea of the sensitivity of this technique, here's actually, these are the trace amounts of zinc that are inside the wood cell wall structure, and there's more zinc in the middle lamella. And so we can use those maps to help us identify where these cells are in the bromine map. And then we basically just defined RRIs with multiple nanodents, and then took the bromine concentration from there. So we calculate both modulus and hardness, but the hardness, for reasons in the nanomechanical cell wall, are more related to the moisture-induced softening of the matrix of the secondary cell wall. So I'm just going to show the hardness results. And what you can see here is that under dry conditions, there's not very much effect of the amount of adhesive infiltration. but under wet conditions, you can see that this adhesive had a quite large effect on the amount of moisture-induced softening that's happening inside the wood cell wall. And so in addition to doing, so we also did experiments with a variety of different molecular weights. And so you can see that these low molecular weight adhesives, they're less viscous and they flow far into the wood cell wall, into the wood structure. And whereas these higher molecular weight adhesives, they stay very close to the cell wall. So really the most interesting thing that came out of this study was that there was a difference between the lower molecular weight adhesives and the higher molecular weight adhesives. So this difference showing that the lower molecular weight adhesive for a given amount of adhesive uptake was more effective at preventing the moisture-induced softening in the wood cell wall. So this is a new insight, but just nanotation and just the cell wall characterization on their own doesn't provide the whole answer. So this is really part of a multi-scale approach that we're taking to really map out adhesive infiltration at multiple length scales. And so we've also at the APS we built a humidity chamber, and we're doing experiments doing a X-ray computer tomography of wood adhesive outlines. And the cool thing about using this brominated phenol is that there's enough of a difference in attenuation that you can use software to actually separate out the adhesive, so you actually really see the adhesive structure inside the cell wall. Then of course we've also got our nanoindentation and fluorescence microscopy. But then in addition to that, we've also started doing small angle neutron scattering to get at what's going on down at the nanoscale. And so in this experiment here, this is unmodified wood. And again, we built a humidity chamber to go into the neutron beam line. And what you see here is these peaks here, these are diffraction peaks that actually correspond to the spacing between crystalline elementary fibrils that make up the cellulose microfibril. And so the cellulose fibril is actually swelling with moisture, and the amount of swelling is actually about 20%, which is about twice as much as the swelling as you see in bulk wood. So this is really a strong indication that if you want to make a good wood adhesive, you've got to stop the swelling inside of these microfibrils. And indeed, what we did was, we took a really low molecular weight phenylformaldehyde adhesive that we labeled with deuterium, and then we put that in the wood structure. And what we saw was that on modified wood dry, you don't see these peaks that correspond to these cellulose microfibrils, but with the delabeled phenylformaldehyde adhesive, you do see these peaks. And so what that means is that this low molecular weight is actually able to get into the elementary fibrils and are stabilizing it. So in the future now we can do studies with different molecular weights, distributions of the phenylformaldehyde, and see is there a higher molecular weight limit for adhesives that can infiltrate the microfibrils, and then if there is, then if you're going to design a new wood adhesive, you've got to have something that can get inside the microfibrils. So that's how we're using this information to make better forest products. Can I take about 5 more minutes, or I'll pull back from lunch? 1 minute? All right. All right, so this is just also some work. So basically, degradation requires ion transport through the cell wall, and ion transport only occurs above a humidity that actually corresponds to what's believed to be the moisture-induced glass transition of the hemicelluloses in the cell wall. So we also design experiments to verify that there is a moisture threshold for ion diffusion in wood cell walls. This is using an arch chamber inside the fluorescence microscopy beam line at APS. You see that low humidities, no ion diffusion; high humidities, lots of ion diffusion. And then getting back to our nanoindentation, you can test cell wall transverse plane, longitudinal plane, and what we actually find is that if you do this at different humidities, when you're testing in a direction that's perpendicular to the elementary fibrils, so you got these softened regions of hemicelluloses, you do actually get a peak that shows up in the tan delta, and we believe that this peak corresponds to the moisture-induced glass transition inside of the wood cell walls. And so this is another tool that we can use that if you want to develop a wood treatment that prevents fungal decay. We believe that you have to target the hemicelluloses to prevent the glass transition, to prevent the ion diffusion, because the initial stages of fungal decay require ionic diffusion in the wood cell wall to do non-enzymatic degradation, to do some non-enzymatic degradation, because there's not enough room in the cell wall for an enzyme to get in there to begin with. So sorry, I went a little over, but I'd like to thank all my collaborators and happy to take any questions.
Do you find when you're using the relative humidity cell that you need to redo the air indent on your transducer as a function of humidity?
So I actually don't have a humidity cell. I have a hose hooked up to the back of my enclosure, and I changed the entire RH. But I do. I always do a new air calibration at every humidity, but I don't find it changes very much.
Okay. Then I don't have to do that study.
Well, yeah.
Yeah, just curious, like when you change the relative humidity, how soon it will respond, the fluid will respond on the saturated?
Oh, it actually, so inside the enclosure takes a while because it takes a while to change the humidity. But like in the, like when I did the X-ray computer tomography, I've done like Littlewood slivers as well, and basically it's pretty much instantaneous. It's not instantaneous, but it's fairly fast. But you saw in the movie that I showed, that was about 2 millimeters by 2 millimeters, and over a period of about 8 hours, it was still changing slightly.
Yeah.
So there is.
I'm curious about that. And
Yeah.
those are a stack of the images and just take consecutively, or
Yep. They were just.
That is it.
Actually, I set those up to run so I could go home and sleep. But yeah, so that started out. It was at like 99% humidity, and then I did a step change to 0, which takes about 1 minute inside the chamber, and then those experiments were collected over a period of about 8 hours. Yeah, I can show you the movie again, and the times are actually on there to get an idea.
Right. Thank you.
All right, thank you.